Hello everyone, welcome to eDufundae. I am your mentor for the topic bacterial cell structure. So let us see what is a bacterial cell structure. With the development of the electron microscope in 1940s, the bacteria revealed themselves as more than simple spheres and rods. That means जब initially इतने advanced microscope available नहीं थे, at that time the bacteria, the structure appeared like of simple spheres. We call them as the coccus bacteria or the rods that we called as the bacillus bacteria. But later on, after the development of electron microscope in 1940s, more diversified and more detailed form of the structure of bacterial cell could be studied. So this is how a normal bacterial cell looks like. It has various organelles inside, it has a presence of cell wall and so on. So let us see the detailed structure. The first part we are going to discuss is the flagella. The bacteria are endowed with flagella and show rapid wave-like movement. So bacteria जब motility show करता है, अगर movement show करता है, that means it is with the help of some locomotory organs, and those locomotory organs are the flagella. Here we can see that the bacteria is showing, here is a single bacterium basically, it is showing the wave-like rapid movement, and these thread-like structures that are arising from the body are the flagella. So what is the advantage of it? The motility is helping the bacteria to move towards the favorable environment or away from an adverse one. For example, ह्यूमन बींग्स, अगर हम एक पर्टिकुलर एनवायरनमेंट में रह रहे हैं एंड वहां पर हमें वो पर्टिकुलर कंडीशंस सूटेबल नहीं है, so we simply walk away from there, right? So this is what bacterium is doing. It is moving away. Now we have the limbs to move, so it has the flagella to move around from one place to another. The bacterial cells have arrangements of flagella, and they can be monotrichous, lophotrichous, amphitrichous as well as peritrichous. Now let us see what are these. In monotrichous form, a single polar flagellum is present. So the plural form is flagella and the singular form is flagellum. In case of amphitrichous, we see a single flagellum at each end of the cell. Then lophotrichous, two or more flagella at one pole of the cell, whereas in case of peritrichous, the flagella are distributed over the entire cell. Now let us see these structures. The first one is the monotrichous and the second one is amphitrichous. So I can see over here that there is a presence of single flagellum in case of monotrichous, and in case of amphitrichous, single flagellum is arising from one pole and from the other pole. So there are two flagella, but they are arising from the opposite poles of bacterial body. Here we have the lophotrichous, that means more than one flagella will be arising from only one pole of the bacterial body, and on the other hand we have the peritrichous, in which the whole body is surrounded with the flagella. Then is the next part, that is plasmids. Bacteria contains small circular double-stranded DNA molecules called as plasmids. Plasmids usually contain fewer genes than the genophore and often confer protective traits such as resistance to drugs and production of toxins and enzymes. That means these plasmids are the genetic structures that are present inside the body of bacteria. They are also capable of self-replication, and they show the resistance to the drugs and production of toxins and enzymes. That means for its own protection as a defense mechanism, it is leading to the resistance to the drugs, as well as it also produces toxins and various enzymes. And this is the benefit that bacteria provides in case of the industries such as medicines, cosmetics, etc. Further, we observe that they may be gained or lost without harming the bacterial cell, and what we are talking about? The plasmids. That means we can gain the plasmids from the bacterial cell without harming the bacteria. Because plasmids can be readily manipulated in the laboratory and transferred from one bacterial cell to another. These are used as the agents that we call as vectors in the modern genetic engineering techniques. So, what we do is in a single bacterium, we see there is a presence of the bacterial DNA, the main DNA, as well as plasmids. The plasmids are also the genetic material only, but they are in the separate forms and they can also self-replicate. They do not require the main DNA for its replication. It can self-replicate. So what the scientists do, they take out the plasmids, make some changes into it and introduce it into the other bacteria for the various studies and for obtaining different results of various vaccines or medicines as well as the genetic engineering processes. I hope you understood the explanation. Thanks for watching.